Yesterday, I gave you that Amoeba Sisters video. I felt like that would serve as a good introduction to DNA replication because this process can be confusing. So the Amoeba Sisters does a great job of giving you an animated animation of the process. And what we're going to do today is we're going to review that process. And while we're reviewing it and I'm drawing it out on my iPad, your assignment is going to be to diagram it yourselves as well. So when we get to that part, I'll remind you, but you can do it on a piece of paper, you can do it electronically, be creative, and label everything. Make sure that you truly understand the process because I feel like that's the best way to learn about DNA replication. So yesterday during the Amoeba Sisters video, you already answered this first question. When does DNA replication occur? We're bringing us back to the cell cycle, right? It occurs in the S phase of interphase. So before mitosis or meiosis can occur, we need interphase, specifically S phase, where DNA replication can happen. What do you think complementary base pairing has to do with DNA replication? So this was something that I asked you 2 different times, I believe. If we're replicating DNA, so we have double helix, right? When we replicate DNA, and we're going to be helicased, right? An enzyme is going to come through, unzip the DNA. So then our DNA is going to look like this, like 2 separate lines. So on one part of the DNA, right, you're going to have some rungs of your ladder, right? Those are your nitrogenous bases, your adenine, thymine, cytosine, guanine. And the other side of that ladder had, those complementary base pairs. When DNA replicates, it replicates in a semi-conservative way, which we'll talk about again in a second. But each strand of the parent strand, which is in the blue, is going to be replicated. How is it going to know how to replicate that strand? Well, there's complementary base pairing. So if there's an adenine here and we're making the daughter strand, then it would know to lay down a thymine. So complementary base pairing is important because that is what allows us to be able to replicate DNA. So just to briefly review some things that you heard already in the Amoeba Sister video that are important for DNA replication. DNA runs in an anti-parallel orientation. So again, that means that one strand is going from 5' to 3', and that the other strand of DNA is going from 3' to 5'. So directionality is referring to the different strands of DNA being referred to the 5' end or the 3' end. So 5', 3' running in that anti-parallel orientation. So the new strand being made is running in one direction while the original strand is being read in the opposite direction. So what that means is if we're looking at the strand that is running from 3' to 5', and we have DNA polymerase making a copy of that strand, the copy is always made in the 5' to 3', meaning that the first nucleotide would be placed here because it's an anti-parallel orientation. So it's making the 5' end, which needs to be placed by the 3' end because they are running anti-parallel. So DNA needs to be made in the 5' to 3' direction. If I'm looking at this strand right here, the one that runs from a 3' to 5', the new strand needs to be made 5' to 3'. That means that the new strand needs to place its first nucleotide on the 3' end because of that anti-parallel orientation. So see how the new strand would be making the DNA 5' to 3'. So it's making it 5' to 3' by placing it in the anti-parallel orientation, placing it at the 3' end of the parent strand. It goes 5' to 3'. The new DNA strand needs to make it from 5' to 3'. So placing the nucleotide at the 3' end to go 5' to 3'. So that is the new strand being made versus the original strand that the new strand is reading in order to make that new strand. Parent strand, so that would be the original, the OG strand, versus the daughter strand, which is the new strand that's being made. DNA is made in a semi-conservative model, which the video mentioned yesterday, and there's a leading strand versus a lagging strand, and in the lagging strand, we have something called Okazaki fragments. So let's just refresh that semi-conservative model. This is a good picture demonstrating what that means. So the parent strand, again, is the starting strand, and in this picture, that's going to be the blue, and the daughter strands are in yellow. So if we look at the parental DNA, double helix, 2 blue strands, the semi-conservative model means that the DNA double helix is going to split, unzip, and each strand of DNA is going to be copied to create what we have down here. One parental strand being intertwined with one of the new strands that were made, one of the daughter strands. So where does the name semi-conservative come from? Half the parental DNA is being bonded with the daughter strand, the new DNA. So half of the original strand gets conserved when DNA replicates. So here are the enzymes that you heard about in the video yesterday. Helicase. I asked you where you think, looking at this, the name of this enzyme, right? Enzymes end in ase. So helicase, what does it do? It unwinds the DNA's helix. So helicase, helix. So that can help you remember the name of the enzyme and the function of it. The video also mentioned gyrase, and that's going to relieve the buildup of torque during the unwinding. So that was the enzyme that prevents the supercoiling of the DNA, right? Topoisomerase is the umbrella term for different, a couple of different types of enzymes. The one specifically for DNA replication is called gyrase. Then we have primase or RNA polymerase. That is what lays down RNA primers. Now remember I asked you what a primer was in your everyday life. So most of you said it's something used before you start doing something. So for example, painting a wall, somebody said you use primer before you paint a wall. Or makeup, you apply a primer before starting your makeup. So that's what this primase, this RNA polymerase primase does. It lays down nucleotides to tell the DNA polymerase where to start. So it's like, hey, start copying over here. It only lays down a couple of bases just to tell DNA polymerase where to start, because DNA polymerase cannot lay down nucleotides unless it has something to start off of. It needs something to build off of. The specific DNA polymerase that is the main DNA synthesis enzyme that lays down and matches the nucleotides and builds the daughter strand, that is DNA polymerase 3. We're not going to focus on numbers of why it's 3 and DNA polymerase 1, why there's no 2, but DNA polymerase 3 is the main DNA synthesis enzyme. It matches new nucleotides to the parent strand or the template strand, the old strand, in order to build the new strand. So complementary base pairing. It's going to lay down the complementary nucleotide. DNA polymerase 1 that's going to replace the RNA primers with DNA. So remember, the primase, the enzyme that's telling the DNA polymerase 3 where to start, it's an RNA polymerase. It's laying down RNA nucleotides. So the bases that RNA uses is going to use uracil instead of thymine. DNA polymerase 1 is going to be able to replace uracil with thymine, also replacing the ribose sugars with the deoxyribose sugars. And finally, the other enzyme that you heard about was ligase. Ligase filling in the gaps of the Okazaki fragments. So remember, when you have this lagging strand in this top picture right here, it's the lagging strand. DNA polymerase can only copy the 5' to 3' direction. So the 3' over here, it's going to have to lay down the 5' nucleotide over here, and it starts moving in this direction to lay down the complementary base pairs. And as the helicase is unzipping the DNA in that direction, right, then it's going to have to, the primase is going to tell the DNA polymerase to start again, so it has to stop, and then it's going to move where the primase tells it to start replicating again, leaving, different separated fragments. This picture doesn't show it yet, but, fragments of nucleotides in this daughter strand. So there's going to be gaps in between them. So ligase is going to fill the gaps. Now, something that I wanted to point out, and if you still need more detail, the slides, again, goes through some more detail for you. So one thing that was mentioned in the video, it looked like the ligase was sealing the gaps by laying down nucleotides, but that is not the case. It fills the gaps by creating phosphodiester bonds. Where are those bonds found? In the backbone of the DNA, right? So remember, double helix, the sugar and phosphate bonds in this backbone of the DNA, phosphodiester bonds, which is a type of covalent bond. Okay, so just something to keep in mind. All right, so what we're going to do is we're going to diagram DNA replication together. So this is something that I do want you to do. It's an assignment, so you're going to finish it for the rest of the class. And if you need more time, then you can finish it for homework. So let's start off by drawing our DNA. So again, your picture should be a little more detailed, or I'm just going to do a quick diagram, but you can add in some bases to better show this. So we have our DNA right, with some complementary base pairs. And what enzyme is going to unzip the DNA? Is going to be unzipped. Here's our 2 strands. The enzyme that unzips it, helicase. So helicase is going to start unzipping our DNA. Now, it's not going to unzip it all at once, right? It's going to create something called a replication fork. And we have a lot of DNA. It's not going to just zip through it all at once. It unzips slowly, which as it unzips the DNA creates this replication fork. Kind of like, when you reach a fork in the road. All right, after helicase, right, what enzyme is going to prevent supercoiling of the DNA as we unzip it with helicase? Well, we need our gyrase enzyme, or topoisomerase. Again, preventing supercoiling. So let's add in, I'm going to erase this now. Let's add in our complementary base pairs. Again, something that you can, put more detail in on your diagram. Also labeling our 3' end and 5' end of the DNA so we know the anti-parallel orientation. We're going to make that top strand the 3', bottom strand is going to be the 5', anti-parallel, so that means that this side is 3', this side is 5'. So how do I read that? The top strand goes from 3' to 5', the bottom strand is going from 5' to 3'. So as helicase is unzipping the DNA, what enzyme do we need to tell DNA polymerase 3 to start? So that's going to be our primase. Primase is laying what type of primase is it? RNA. So it's laying down a couple of nucleotides, telling DNA polymerase 3 to start. Where to start, right? So primase lays down our primers. And again, why did primase lay them at the 3' end of the parent strand? Because DNA polymerase can only make the daughter strand in the 5' to 3' direction. So this daughter strand, in order for DNA polymerase to make our daughter strand, it needs to lay its first nucleotide at the parent strand's 3' end. So it's laying the first nucleotide, the primase is laying it at the 3' end of the parent strand, so that way DNA polymerase can make the new strand from 5' to 3'. So primase laid down the starting nucleotides, which is going to tell DNA polymerase 3 where to go. Now, also needs to do it for the bottom strand. So again, needs to make it in the 5' to 3' direction. So primase is going to lay down the primer, telling DNA polymerase 3 where to start. Now, this bottom strand is going to cause an issue, right? But we'll talk about that in a second. So now that we have primase telling DNA polymerase 3 where to lay down the nucleotides, this top strand is easy. As helicase is unzipping the DNA, the daughter strand that's being laid down by DNA polymerase 3 is still going in the same direction as DNA is splitting. because it's making it from the 5' to 3' direction. So this strand is the leading strand because it has no issues. It's going to be able to create that daughter strand easily all the way through. It only needed primase at the very beginning. Easy. This bottom strand is what's going to give us a problem. This is called our lagging strand. Why? Because it's lagging behind. Because this strand is being laid down from 5' to 3', but DNA is unzipping in this direction. So DNA polymerase 3 still going to lay down the nucleotides after primase told it where to start, right? But now the DNA is still unzipping. So primase is going to have to, I should have made this longer, but let's pretend that those are also on the strand. As the DNA keeps unzipping, primase is going to have to come back, lay down some more primers telling DNA polymerase 3 where to go again, and then DNA polymerase 3 will lay down some nucleotides, but the DNA is still moving, so then it's going to leave a fragment because it needs to move along with the DNA as it's unzipping. And these fragments, see how there's, space between them, right, where there were no nucleotides placed. That is called an Okazaki fragment. So what enzyme is going to connect these Okazaki fragments? That would be ligase. So ligase fills the gaps in between, oops, that wasn't one of them. Oops, but here's one. Fills the gaps in between these Okazaki fragments. It fills the gaps by phosphodiester bonds, okay? And then finally, DNA polymerase 1 will go to the primase and change the RNA primer into the RNA bases and nucleotides into DNA. Ding it down here. Okay? So make sure, diagram that out so you can truly understand what's taking place. And as always, please let me know if you have any questions while diagramming.